Okay, welcome everybody. So we're really pleased to come together this morning to take a journey from a molecule and a drug all the way from target validation through discovery and characterization. It's been a nice experience pulling the whole seminar together. We're bringing together today G.CLIPS, Twist Bioscience, Twist Biopharma, and also IPBS. These are three groups that work along the progression of the story. And so we thought today we would do something a little bit different and organize ourselves walking through time and seeing how the molecule progresses from one state to another. And we'll even be passing back and forth between ourselves in order to continue to follow the story in a way that makes sense. We're going to go ahead and kick off. We seem to have sorted out our sound and video issues. So welcome everyone. We're really pleased to have everybody both here in person in lovely Toulouse and also our friends joining online. If you could just hold your questions until the end of each session, then we'll be happy to answer those and then we'll progress on to the next. Mehdi?
Hi, everyone. Very nice to be here with you today. It's a nice agenda, as you can see here. So we will start at 9 with Dr. Rosie Dawalibi for a topic about changing paradigm on membrane protein as the targets. And then we will follow up with Dr. Radha Parma from Twist Bioscience for the future of biologics with an integrated offering of immunization libraries and machine learning. Then will be a short coffee break, very short. Then we will follow up with binder pharmacological profiling by J-validation assay, and then a quality control and characterization by Julien Marcoux from IPBS. And we'll, last but not least, speak about quality control and characterization of monoclonal antibodies and ADCs by structural mass spectrometry, by Dr. Julien Marcoux. So there will be a closing remark at the end, so keep your question at the end. Okay. So let me first start with Rosie. Let me introduce to you Rosie Dawalibi. She's the CEO and co-founder with me of G.CLIPS Biotech. Rosie holds a PhD from the University of Lausanne and a postdoc graduate from Stanford University. After several successful experiences in several biotech companies, she founded G.CLIPS Biotech on a perspective of membrane protein as a target, her main research and goal over the last 10 years. So, Rosie.
Thank you, Mehdi. Thank you, Jessica. Thank you, everyone, for being here today. I'm really pleased and honored and humbled to be with you today. It's a great pleasure, actually, to share a little bit of the experience I got through these years with you today. Let me just start by sharing the presentation. I hope everyone can see it. And everyone can hear me as well. So, the topic of today will be changing paradigm on therapeutic targeting membrane protein. And the idea is to walk you through our vision, the vision that G.CLIPS has on how to treat membrane protein as drug targets. And what we want really to point out here is really that environment matters on this membrane protein. And you'll see that one membrane protein cannot be just one target, but many different targets. And so that's the aim of today's talk. So to start with, what I want to tell you is that actually behavior in general, our behavior depends on our environment. So of course, a very nice team as we have at G.CLIPS can be in different environment, and its behavior, as you can see in these pictures here, can be very different. And so the way we act and behave is different in a lab, in a team building, or in a congress or a meeting. And actually, it's the same for a membrane protein. The behavior and the way this protein will function will be very different depending on the environment in which these membrane proteins are. So, for instance, the membrane protein can be a same membrane protein, the same target can be expressed in different organs, as you can see here. So it is known that basal activity and signaling of the same membrane protein can vary depending on the tissue in which it's expressed and what are the effectors that are around this protein. Also, some receptors or membrane protein or surface marker can be expressed both on immune and tumor cells. And their affinity towards their ligands and their signaling, they can be very different and they can vary depending on the tumor microenvironment versus the healthy or the immune cells. And similarly, some membrane protein, like some G protein coupled receptor, can signal from different cell compartments. And in this case, as you can see, each of these signaling pathways is different and can trigger different cell responses and phenotypic answers based on where they are signaling from. So why are we interested in membrane protein in general? So these membrane proteins are really targets of huge interest with a lot of challenges at R&D stages. And as you can see here from this picture, actually they are the target of more than 50% of all drugs that are approved by the FDA, meaning that more than 60% of drugs that we can take have as target a membrane protein. So they are really of huge interest in many different fields and in many different indications for antibody discovery. But one real bottleneck working with this membrane protein is the quality of purified antigen and membrane protein, specifically when you want to use them in antibody discovery. But they are really, this bottleneck is really important because it can actually affect your discovery at many different levels, at hit validation state, hit to lead, even before preclinical and clinical studies to understand the mode of action and to do the target validation. So it's really of a huge interest to have a good quality purified membrane protein to be able to assess all these steps. So coming back to the environment, I wanted to show you here that membrane protein environment has been shown to modify their structure and function. So I took 3 examples here. So you can see in the first example that you can see here that ions that are in the environment surrounding a membrane protein can really play a role and act as positive or negative allosteric modulator for many GPCRs, especially the ones implicated in CNS. So when you work with purified protein, it's important to take into account, for instance, the ionic strengths that might be important, considering the target-specific pathology and tissues. Lipids are also of very, very important component for stabilization and for the structure and function of the membrane protein. Again, here we have an example where lipids, you can see, so this is what you see here is actually activation of a receptor with surrounded with different kind of lipidic bilayer with different lipids. And you can see actually that when you add the agonist, you have different kind of activation, as you can see here. And when you add the antagonist, you have inactivation, but in very different manners. So the efficiency and efficacy of each in each of these membrane is quite different, meaning that you can really have a very different behavior or effect of each of these molecules, depending on what kind of lipid surrounds your protein of interest. And finally, very interesting thing is that pH also can play a crucial role depending on what we are looking for or targeting for. For instance, it was known that low pH and the tumor microenvironment acts actually as a kind of a shield for tumor cells. And so targeting tumor cells has then to consider drug recognition, recognizing tumor cells at low pH. So that being said, G.CLIPS' mission is really to uncover a new class of therapeutics targeting membrane protein to treat disease with high unmet medical needs, but with a new vision. So G.CLIPS' vision is to radically change the old paradigm of membrane protein to discover a new class of therapeutic. So the old paradigm would tell us that one membrane protein is one potential target, and so one potential candidate. And so you would select with one purified membrane protein in some sort of structural or folding or conformation and environment. And then you will get candidates that are not always selective, and you go to a lot of fail at preclinical and clinical stages. And so that's really a problem because the potential there is potential therapeutics in that cases that are missed and not selected. And some of the selected candidates will be very poorly selective in vivo in the testing. So G.CLIPS' idea is really to change this paradigm and say, okay, we have one membrane protein, we're gonna extract it, but we're gonna extract it in different conformation. We're gonna embed it in different kind of lipidic environments with specific membrane composition and specific ion strengths and pH surrounding it. And from these, we're gonna select for a specific antibody or small molecule, but here, of course, our interest is in antibodies that is really corresponding to the protein in its physiopathological conformation and environment. So the idea is to have from one membrane protein several targets, and so several candidates that will have different effects in different indications, and thus have new families of therapeutic with increased indication-based selectivity and improved therapeutic windows, and of course, decreased off-target and off-tissue effects. So for that, G.CLIPS can do two things, or can deliver basically two things. First of all, we can generate the new targets stabilized in physiopathology-like conformation and environment in different formats, such as bicells with J-mixes and nanodiscs. And these can be already so the membrane protein purified and stabilized in this desired and specific environment for that it can be used for immunization and antibody screening, in vitro assay development, etc. But what's really unique about our technology is that we couple this with, we associate it with a functional testing. So we have a functional testing based on fluorescence that allows us actually to look at structural changes that happens into the target and then follow the activation or inactivation of a specific membrane protein. And this can be very useful, first of all, to make sure that what we purified is functional, of course, but also to do all kind of validation, going from target validation, mechanism of action profiling of drugs, and lead validation and profiling of candidates. So this is what we can propose, and to do so, we use actually our unique platform, the G.CLIPS J-Screen platform. It's a platform that integrates target expression, stabilization, and validation combined with the profiling of the drug candidate. And as you can see, our platform is based on 4 important assets, proprietary assets that we have at G.CLIPS. First of all, we have our J mixes. So those are proprietary mixes that allows us actually to extract from the membrane the protein of interest. The mixes are made of detergent, of course, because we need to extract them from the membrane, but also all kind of other components like different ions, different pHs, lipids, surfactants, that allows us, first of all, to stabilize and also to block somehow the receptor or the membrane protein in a specific conformation. And then we can use our selective screening assay, the G.CLIPS assay, that allows us actually to choose a specific J mix that would stabilize a specific conformation. So we can have the same target in different conformation based on the functional assay that we have, the fluorescence assay that we have, that allows us actually to check for this conformational changes and see at basal level which conformation is stabilized. We have also our G-lipidomic database. So this is the lipidomic database in which we have the lipidic composition of many different tissues and cell types, in which actually we did mass spectrometry to assess the composition of these membranes. And so this database allows us to reconstitute the purified protein in a specific bilayer or on a bilayer that allows us to, let's say, mimic the tissue of interest that we want to target. And finally, as mentioned, we will use our validation assay to do a different kind of validation, target validation, but also candidate profiling, as you will see in a few minutes with the case studies that I will present to you. So here I depicted actually how G.CLIPS enabling technology can help producing new antibody therapeutics targeting membrane protein. So this is a little bit the workflow that we can do at G.CLIPS and with some of our partners. So first of all, G.CLIPS will produce the protein in mammalian cells to be able to have enough material to then use the J mixes to extract them from the membrane. Then we will have the stabilization in specific conformation via the J mixes and the G.CLIPS assay. As you can see here, actually our fluorescence assay is based on a shift in fluorescence based on a structural change. And from here, actually, those stabilized protein antigens and J mixes can be used for immunization, for instance, but it can also be used for library screening, as can be with some of our partners, with Twist, for instance, and some of the projects we have together. And so from there, our partners can use it for immunization or library screening. And then we have selected hits that can be then verified and validated once we have reconstituted also these protein in specific tissues. The idea is, of course, to take the tissue of interest or of the cell type or the tissue that we want to target, but also to reconstitute the protein in some other kind of environments where the protein is naturally present, to do selective screening and eliminate like the antibodies that would recognize equally well the target in all the different environments and select for the ones that recognize much better in the tissue of interest. So once we have hits that are validated, we can, of course, profile them using the G validation assay, but not only, because of our presence at the IPBS, we have access to the PICT platform, represented here by Virginie, that will talk to you a little in a while about what they can do. And that's really nice because we can not only verify via G validation assay, but also do some orthogonal validation using other kind of assays like a spectral shift assay and some other assays. And so finally, once we have validated those hits, we select for a lead candidate that recognize the pathology-like target only with a desired mode of action. So at G.CLIPS, we've been working on really different or diverse kind of membrane proteins, going from receptors in monomeric form or dimeric form, homodimers or heterodimers, of course. We worked with adhesion proteins, also in oligomeric or monomeric form. We worked with cell surface markers. We worked with challenging protein, like big ion channels and so on. And the challenges stays always the same, is to stabilize the protein in vitro, master their conformation, to be able to select for a candidate with a specific mode of action, and then select for the desired environment and validate the target and profile the candidates. So now I will talk to you a little bit about the case studies that we have. So part of this case study will be shown here, but the other part will be after Rada's talk. So stay tuned, come back to hear more about all of it. So the second case study that I will be talking about is a joint effort with Twist Bioscience, which, by the way, are very, very, very nice people to work with, great partners. And so in this partnership, so we have different projects ongoing with Twist. I'm going to just say a word about the C5AR project, just to show you how the antigen was produced and how then Twist used it for their lead validation and characterization. And so the aim of this project was to produce antigen that allows our partner to do antibody validation. And then we also did ourselves some pharmacological profiling on some of the antibodies that they were sent by Twist after they validated the leads. So just a quick word about C5AR. So this is a very interesting, actually, target in inflammation, but also in different indication in oncology. So it has been, so it's a receptor that is part of complement receptor family, and it's also implicated in the inhibition of non-immune cells inflammation and maintains the epithelial integrity. And so it was shown that there's a synergic effect, actually, when you block the complement receptor or the complement pathway, together with other strategies that are used to treat cancer, like blocking of some immune checkpoints like PD-1, or also when you do both complement signaling blockade and some chemokine or chemotherapy combination. And the last combination that has been shown to be interesting, using the complement is also as a clinical use with a PD-1 antibody. So again, the workflow that has been used by G.CLIPS is quite similar to the one before. So first of all, expression of the target, then extraction and stabilization, lead antibody validation at Twist, and then antibody characterization at G.CLIPS. So here in that case, since we didn't need a huge amount of the protein, we decided to go for transient cell production and transfections. So the receptor C5AR was produced in mammalian cells, of course, but through transient transfection, and we had very nice expression at the cell surface. So our flow cytometry histograms are always done on non-permeabilized cells to detect, of course, the extracellular or the target that is on the surface. In this case, we use the anti-flag antibody that is on the N-terminal end of the protein, so at the extracellular domain part, to detect the protein. With also, we have a control with irrelevant antibody and with non-transfected cells that are also marked with the anti-flag antibody. So the second step was to use our J-mixes again to purify, extract, and stabilize the protein from the cell membranes that we have prepared previously. And we decided actually to go for 3 different lipidic compositions. First of all, to test what kind of basal activity we will have in each case, because in that case, it was really interesting to have different conformations that can be stabilized. So we used mixes with different ionic strengths, but also with different lipidic compositions. So the first thing that you can see here is an ELISA that we have done with the protein that has been extracted with different kind of J mixes. And you can see already that the recognition with anti-flag is not the same, meaning that the extraction efficiency also is not always the same in each of these mixes. But that was just a quality control to see that we really extracted the protein in each of these conditions and that we can code them to use them afterwards in efforts of in vitro assay characterization. And so the protein also was, so from these J mixes, we decided to choose one. Here I show you the example of one, which is the sp3L10, and we decided to decided to do large-scale purification on this antigen, and of course Western blot validation, and then stay tuned because the rest will be coming soon. After Rada's talk, who will show you a little bit also about what we have been doing with this. So thank you for listening to me. And so in a few minutes, Rada will tell you more about how Twist, all the Twist tools that they have, and also a little bit of what kind of characterization has been done with this receptor. And then I will tell you a bit more about how we validated the C5AR, and I will talk to you also about another case study that we had on a chemokine receptor. Thank you very much. And of course, if you have questions, I'll be thrilled to answer them. Discrepancy between the genes that we use. Yes. What you consider as the natural environment. Sure. So I'll just repeat the question because I'm not sure that people that are online can hear it. So your question is if we have discrepancies between sometimes J mixes that we use and that are the best mixes that we can see, and they do not maybe correspond to what we think is the natural thing to go with. And actually, that's a very good question. And in general, what we do is that we purify, in that case, the protein with both environments. And then we do a series of benchmark testing. So we use G validation to validate the antigen that we have purified, either in our best mix or in the one that should be more physiopathological. And then we test our benchmark ligands. So either the natural ligands or sometimes it's a standard of care that is usually used or so on. And we see then if ours is best or the other one is best. And most of the time, there's no really best. It will depend on what kind of mode of action you want to go with. It would depend on what kind of targeting you want. So for instance, we had once we had this kind of dilemma, let's say. And Actually, it depended a lot on the mode of action of the drug that you were testing. So for agonistic drugs, our J-mix was best, but for antagonistic or blocking drug, the natural one was better. So it really depends on what you're looking for in that case. And in that particular case, they were looking for an antibody that would block the protein or antagonize the protein. So we went with the natural one, but it will really depends on what you're looking for as a drug candidate.
I have a similar question, I guess, the environment. So for your first case study on the clusters, so you said clusters and not oligomers. So I was wondering how well they were characterized. And my question is related to the size of the nanodisk. I was wondering if you could try different sizes and if this could constrain somehow the oligomer and then the recognition by the antibody.
That's indeed what we did. So the question is for the cluster case study, I said clusters are not oligomers, of course, but we actually made sure, because we tested on with different antibodies that would recognize. You see on the scheme that I have depicted here, you see that we have several domains. So if it's just oligomers or precipitating things or something, you won't be able to detect each of these. And we had the chance to have antibodies against at least 3 of these domains. And that's why we verified each time that what we have purified is recognized by each of these antibodies. And then the size of the nanodisk, of course, we used different belt proteins for clusters and monomers because it's really important to make sure that the clusters can go through the nanodisk. And we wanted also to be sure that when we have monomers, they do not go in oligomeric form into the nanodisk. So yes, we played with the size of nanodisk to have more or less space for the clusters or the monomers to be reconstituted.
Even for the clusters, you see that if you use different sizes, you will have different number of aggregate or clusters, and
it's possible. So these were tested. The reconstituted HDL particles were tested via DLS, and it was really homogeneous. So I think they have favorite numbers of protein that comes together, but I would guess it would really depend on the target. If you have a target that can be in dimers, trimers, etc., you might have really a selection of nanodisk.
Thank you, Rosie, for the talk. My question is a bit of naive one, but for one project, one university, how much time is there between the beginning and the end of your workflow, and which steps are the ones that are the most painful?
Sure, thank you, Stella. So the question is, what is the length of a project from project design to the end of a program? Actually, well, it depends on the target, of course, and how difficult it is. But for some of the receptors that we really now have very good experience with, like G protein coupled receptor, or even with the cluster, it can be quite fast. So minimum time to go from target production to validation of a candidate can be like 3 to 4 months. But for more complicated ones, like the cluster project, it was a bit longer because also they needed to do the immunization, and we needed to send the antigen each time. So it was a bit a longer project. So it depends on the project and the project needs. But let's say from a classical project, from production to validation would take around 3 to 4 months. Yeah, so to quantify the size of the nanodisk, we usually do a DLS, so the dynamic light scattering, thanks to picked at the IPBS. So that's one of the, let's say, the advantages of being here at the IPBS and having access to this kind of material. So yeah, we even do either do the dynamic light scattering, or sometimes we even go to SEC, but it's, I mean, it gives you an idea about the molecular weight of your nanodisk, but it's not as precise as DLS, so I kind of prefer doing DLS. And for affinity binding, you mean between the antibody and the receptor, for instance, and so on? Yeah, so either we do classical dose response ELISAs, and we calculate the KD, and this is something I'll be talking about in the last case study. Or we have now also the dynamic, spectral shift assay that we can use. It's a nanotamper machine, I think, Virginie, if I'm not mistaken. And so we can use it as well to do a series of dilution and dose responses using the, but Virginie will talk about it better than I. So yeah, you can use it to follow the spectral shift and do those responses. Online, if you have any questions, I'll be happy to answer them. You can always write them on the chat. Okay, so if there's no questions online, I will leave the floor to Jessica, who will present RADA. I've just opened her presentation.
All right, great. Thank you, Rosie, for a really a great start here to the seminar. I can also just reiterate what Rosie said is absolutely true. Of course, there's a natural fit, literally and figuratively, between an antigen-producing group and a group that specializes in antibody discovery. But it's an additional joy and improvement to the whole working experience when the human beings within those teams also work well together. So it has been a really nice experience to work together. Our next speaker today is Radha Palmer. She's from Twist Bioscience, and she's our technology and science consultant. Radha came to us from GSK, where she has 15 years experience heading their antibody engineering team in the drug discovery space. She has experience taking projects through from target validation, so clearly this is quite up her alley today, through to candidate selection, and has also worked on projects that have progressed to the clinic and then have gone to market. So a success story, which is great. She's an invaluable resource for our partners and our conversations. I work in business development, and she and I work together to interact with our incoming partners and ensure that we have the right type of input and scientific direction to bring those projects through to fruition. And she's additionally listed as an inventor for an antibody, which Blenrep, yep, that has gone to market. So that's the one. And so with that, I'm happy to invite Radha up to the podium.
Yeah, it's very nice to be here in Toulouse, and thanks very much for that introduction, Jessica. Yeah, so today what I'm going to do, I think Rosie has set the scene really nicely with her talk there. I'm going to tell you a little bit about Twist, a little brief overview, and then I'm going to focus in and tell you about how we at Twist Biopharma Solutions are really writing the future of biologics with this integrated offering of immunizations and libraries and machine learning. And I've got this case study on C5a receptor that I'm going to share with you, where we've actually used such an integrated offering. And as part of that offering, I'm going to just highlight exactly, you know, how we've worked with G.CLIPS and the antigens we've used there. So just to make sure. Right. OK, so Twist, we're a DNA synthesis company. We make DNA. We make DNA using the same phosphoramidite-based chemistry, the conventional way of making DNA that everyone else uses. But what's different is we've really miniaturized that chemistry down onto a silicon platform. And the silicon platform is roughly the same sort of footprint as a 96-well plate. So where everyone else is making an oligo per well of a 96-well plate, our miniaturized process allows us to make 1 million oligos on the same sort of footprint. And we can build these oligos with base by base precision up to 300 nucleotides in length, although we're pushing that length as well, so that's coming. But essentially, by really miniaturizing this process, we're using very little raw materials. So it's a very cost-effective process, and also a very green process. And we're quite proud of the fact that it's so environmentally friendly. So having such low raw material use and an effective, efficient process means we have very little waste, and it makes it a green process. But the other thing to say is that because we're using this silicon chip platform, we have very good control when we're making these oligos, and we have industry-leading quality. So really the things to note about Twist DNA is that we have 100% oligo representation, so no dropouts, extremely high uniformity, as shown by that plot on the left, and an industry-leading low error rate of 1 in every 3,000 bases. So it's high throughput, cost-effective oligos with this industry-leading quality that we then use as the starting point to build some custom products and offer some services. So what can we do? Well, we can assemble the oligos together to make gene fragments. We can clone those gene fragments into our vector, your vector, to make clonal genes. If it's antibody genes, then we can take this a step further now and provide you with milligram quantities of purified antibody. We can elute those oligos in various ways off that silicon chip to provide you with really high quality oligo pools. This is useful for things like perhaps guide RNA for CRISPR screens. But essentially, we're using these oligo pools as the starting point to build DNA variant libraries. So the genes, the antibody production, the oligo pools, and the libraries, that forms our synthetic biology division at Twist. That's our products arm. But essentially, we're using these products within our discovery services as well. So we have a discovery services arm. We also have an NGS arm and a data storage arm. But I really want to focus in on the discovery arm here. So this is what we call our Twist Biopharma Solutions group. And really, within Twist Biopharma Solutions, we're able to provide this comprehensive end-to-end antibody discovery service, where we work with clients and partners who come to us with their target of interest that they wish to discover antibodies to, and a wish list of what they want those antibodies to look like. So they want this particular affinity, they want certain species cross-reactivity. They want to ensure that we're binding very specifically to the target with those antibodies and not to any closely related family members, for example. So the client and the partner will bring us their target and their wish list, and essentially we can carry out the entire discovery workflow to deliver development-ready drug candidates. So we could carry out some repertoire generation. We have a number of different discovery platforms that we can use to identify a set of hits that we can then really characterize in a high throughput way, screen and characterize to identify a set of leads. And if required, we can even optimize those leads and validate those leads further. So this is what our ability is. We don't always work with our clients and partners to carry out the entire discovery workflow. We have a very flexible way of working, so we can pick and choose, what the client requires and carry out parts of the discovery process as well. And just to home in on the discovery services that we offer, the discovery platforms, we have a number of different platforms that all sit within this Twist Biopharma Solutions group. So we have what we call in vivo discovery options, in vitro discovery options, and in silico discovery options. And sometimes we're using these individually, but more and more so we're using these in combination for getting a better chance of success. So within our in vivo discovery, we can carry out immunizations of various animal species. We have a number of different immunization strategies that we can use, and we have expertise with using the beacon workflow for single B cell sort and screening. And I'll tell you a little bit about that in a minute. With our in vitro discovery, we have real expertise in phage display, and we have sitting on the shelf a number of different antibody discovery libraries that I'll tell you about. And on the back end, we have a real ability to carry out high throughput screening and characterization. And with our in silico discovery, we have a team of data scientists and bioinformaticians at Twist. We're building these kind of deep learning models and language models. We've got models in place that assess developability, and we're really using AI/ML for humanization and optimization as well. But we also work very collaboratively with experts that are external to Twist in this space as well. So just to give you a little bit more detail on our in vivo discovery. So as I mentioned, we can immunize different animal species. We can immunize alpacas for VHH discovery, mice, humanized mice, and we have our own proprietary hyperimmune mouse. So this is a mouse that we've genetically engineered to have this broken immune tolerance. And that makes it easier for us to mount an immune response against those kind of less immunogenic regions of the target. So we get this kind of broader epitopic diversity, a greater total sequence diversity, and really we can get these antibodies that are cross-reactive to those high homology targets, something that's high homology in mouse. And if you look at the bottom left here, you can see that as we go up in percentage homology to mouse, we are easily able to mount these immune responses with the diversimab mouse compared to some of the other mice species there. And so we can end up with these mouse anti-mouse cross-reactive antibodies, right? And on the bottom right, it just gives you some data that's come from our beacon screening, comparing the humanized mice to the diversimab mice. And you can see that not only do we get an increased number of hits, we are also able to find these triple cross-reactive clones, ones that bind to human, cyno, and mouse target with our diversimab mouse. And regarding immunization strategies, we would essentially look at the targets that the partner's bringing to us, and we'd pick out what we think would be the most successful immunization strategies. But here are just some examples of what we're able to use and what we've used successfully. So for things like difficult to drug targets, like GPCRs, that need to be folded correctly, as Rosie kind of mentioned, we are finding that we're getting success with DNA immunizations, but we also have this method where we can essentially carry out subtractive immunization, where we tolerize the animals initially with the parental cell line prior to immunizing with the cell line that expresses the target. And sometimes on campaign 3 here, you can see that if we're not using the diversimab mouse, we have other tricks to ensure we can find those binders to high homology targets where we've got our own proprietary adjuvants. Okay, so this just shows you the basic workflow for our in vivo discovery. And essentially we can take two paths. We can either take the path of using the beacon-based B cell screening approach, where we can sort individual B cells after immunization and tissue harvest, and then carry out a set of assays, screening assays on the beacon to obtain sequences. Or we have got our rapid high throughput hybridoma process, which takes longer, but is still quicker than most hybridoma processes, where we'd carry out fusions and then primary and secondary screens, and then characterization prior to receiving sequence. But I just wanted to highlight a little bit more on the beacon and just tell you the advantages that this platform has, really. So the first thing really to note is that it's extremely high throughput. So we can actually screen 100,000 B cells in a day on this. And we're able to carry out a number of different assays in parallel and sequentially in the space of a day to really build a picture on what we think are the most promising hits, which we can then export and work with. And the assays can be very customizable. So as long as they have some sort of fluorescent readout at the end, we can really build the assays up. And the other thing really to note about the beacon is the speed. So from project start to getting those paired heavy light chain sequences of the most promising hits can be as little as under a month, actually. So this is really just some slides which show how it works. So there's a chip onto which the B cells are loaded using light. The B cells are loaded individually into each of these pens. And once the B cells are within the pens, what happens is the antibodies are secreted, the IgG is secreted up into these channels that are above the pens. And essentially, we can carry out a number of assays here because they all have this kind of fluorophore readout. We'll get blooms appearing on above certain pens. And those are telling us something about those particular B cells. So that helps us build this picture and identify the most suitable hits, which we can then export onto a different plate using light again. And then from there, we can sequence the variable genes in a paired way. And just to talk about those assays that we could carry out on the beacon, as I mentioned, there's quite a lot of flexibility and quite a lot of experience within the team to set up multiple assays and run them. So one of the things we can do is look at cell binding. So within those channels in the beacon, we can very homogeneously and uniformly fill those channels with cells that are expressing the target and therefore look for binding to cells. And then the teams actually managed to find a way where they can homogeneously mix two different cell types and load those uniformly in the channel. So in fact, we could get binding to cells with two different kind of targets, if that's of interest. The other kinds of assays that can be run are ligand blocking assays or receptor blocking assays, competition assays, neutralization assays. These can all have these fluorescent readouts and can be quite easily set up. We can multiplex. So if we're looking at cross-reactivity, we could essentially label each of those different species of target with a different fluorophore and actually in one assay identify, which antibodies are triple cross-reactive, for example. And the team, because they've been working with the beacon for a long time, they've gained this experience and they're able to set up even some functional assays on the beacon. So the team's routinely doing these kind of reporter cell line assays to look for agonists and antagonists. And they're also developing currently this internalization assay. So quite a useful set of assays. And there's quite a lot of flexibility to bring assays on board. But just to give you an example of how we've used this internally. So a partner came to us with the target of interest and quite a long wish list. So the partner wanted to ensure that we found antibodies that bound to the protein target as well as the cell express target. The partner wanted to ensure cyno and murine cross-reactivity, ligand blocking activity, and also wanted some idea of how they compared against a benchmarking antibody. So the team were really kind of able to design this beacon screening strategy with, parallel and sequential assays to essentially run all these assays and then export a number of hits at the end of a very long day. And they were able to kind of export 274 hits for this particular project, of which 47 actually met all of what the partner was looking for. So quite a lot of success there. I'm just going to move on. Final slide, just to summarize on the in vivo discovery. The team does have a lot of experience combining that Diversimab mouse platform with the beacon. And this slide really shows the campaigns that we've run using this approach. So each of those different verticals is a different campaign. And you can see the number of hits that we're getting through per campaign. And, you know, with this affinity range, which is really within the desired affinity range. So quite a lot of experience with this platform and quite a lot of success as shown on this slide. So coming back to these multiple discovery platforms, I just want to tell you a little bit more about what we can do with our in vitro discovery. And really the power of what we have here at Twist is our ability to build these high quality DNA libraries. And that's really what we've done in terms of different antibody discovery libraries here. And it starts with actually how we're making DNA on that silicon chip, because we're able to have this base by base precision when we're building these oligos. We can have a lot of control when it comes to, you know, constructing and designing libraries. So there are a number of different ways that people tend to have libraries built. You can go for the degenerate approach using NNK or NNS oligos, or a trimer-based approach where each amino acid is encoded by a single codon. But what happens with these other approaches is that the constructed library can be quite a distance away from the design library. There's some biases that are introduced. There's less control on what you end up with, because you can't control for stop codons or cysteine residues being incorporated or certain restriction sites being included. Whereas with a Twist library, because we have that ability to build each of the oligos as per the design with base by base precision, we can really include the things we want to see within the library, certain codon usage tables, certain ratios of amino acids at particular positions. And we can also exclude what we don't wish to see. So cysteine residues or liabilities like isomerization sites, deamidation sites, restriction sites, stop codons, etc. So it means that the constructed library is so close to the designed library, and it means that you end up spending less time really screening that library, and because you'll find more of your variants of interest much more quickly. So essentially, our ability to build these high quality libraries means that we've done just that for our in vitro discovery. We have 20 or 21 libraries kind of sitting on the shelf ready to use for phage display. And, you know, we have these on different formats. We have Fab libraries with a common light chain. We have a number of single chain FV libraries. I think we have about 6 VHH libraries. They're all built in a slightly different way. Some are naive, some are target class specific, some are based off of natural human repertoires, some are more synthetic. They all come in a little bit differently in terms of how they're built. But what we can essentially do is take the partner's target, identify the most suitable libraries that we have sitting in our library of libraries, pool them if they're all of the same format, and then run them through our panning and screening arm. So we have a number of capabilities within panning. So we can take cell-based panning, we can carry out cell-based panning or bead-based panning. We can use different antigens here. I've got some data in a minute just to show you how we've used the G.CLIPS antigens at this stage. You know, virus-like particles, other nanodiscs. There's a whole host that we can work with at that stage. But the team is really experienced in designing these panning strategies. So yes, the quality of the antigen that is really critical to ensure a successful campaign, but also the experience in trying to design a panning strategy that's really going to answer all those questions and tick that list off to ensure we're getting those antibodies as per the wish list of the client is really important as well. So with design a panning strategy, we tend to do multiple rounds with increasing stringency. We're looking for enrichment. We're also carrying out some NGSQC on the later rounds of panning and using all that information, sometimes with some phage ELISAs if it's a soluble target, we can then select a number of hits that we can then reformat. So we reformat to IgG or VHHFC, or sometimes single chain FVFC. And then we can just plug this into our antibody production at Twist. And I just want to highlight that because that's a standalone product. So sometimes we have people doing their own discovery and then just dipping into our antibody production system here. So essentially what we do here is we take, say, variable region sequences. We will synthesize the oligos necessary to build those genes. We'll clone them into mammalian expression vectors that encode the constant regions that the client's after. And we can run some transfections using generally HEC293 cells. We are starting to offer CHO out as well. And essentially, it's kind of a plug and play system on the Hamiltons, where we can then even purify using protein A and G on the Hamiltons to produce kind of milligram quantities of antibody. So we have different scales. We have a 1 mil scale and an 8 mil scale. The 1 mil scale transfection generally provides about 150 to 100 micrograms of purified protein, whereas the 8 milligram scale can provide about a mg of antibody. And so you've got this purified antibody. We'll give you some basic QC, a digital gel, just to give you an idea of size and percentage purity, and A280 reads to give you an idea of yield. But we can deliver those to you, or we can take it a step further. And there's a whole host of different assays that we can do on the back end. So more QC on those purified antibodies, size exclusion chromatography, a whole suite of developability assays. DLS is something we do on our antibodies, hydrophobic chromatography. There's a whole range of assays we can do on developability, but actually what most of our clients and partners wish for us to do is actually check binding to target. And so we have ELISA set up for that, but mainly what's of most interest is our high throughput abilities in SPR. So we have a number of CAR-Terra systems in a row, and we can really give on rates, off rates, overall KDs, and rank hundreds of antibodies in terms of affinity ranking, and then actually take some forward for epitope binding on the CAR-Terra too. Then we have an ability to carry out some flow cytometry if we're looking at cell binding, and we can also carry out some functional assays on flow cytometry too. So we have a lot of kits. We can do quite a lot on the back end of these antibodies as well. Okay, so really, by having these many different kind of discovery platforms all within this one group, Twist Biopharma Solutions, what we can start to do, what we're doing now is really taking those combined benefits, having multi-pronged approaches to ensure that we're actually being successful with our discovery campaigns. And this is proving to be really useful, especially for those difficult to drug targets like GPCRs. And, Rosie's already talked to you about C5a receptor. It's a target that we thought we'd test out this multi-pronged approach on. This target is of particular interest because actually to date, there aren't any approved biologics that block this particular receptor. So we had particular interest in it, especially in the immuno-oncology field. So this is what we picked, and we used as a case study. We tested out this multi-pronged approach. So what did we do? Well, we did some in vivo discovery, some in vitro discovery, and some in silico discovery. With the in vivo discovery, what we did is we immunized the diversimab mouse that I talked to you about. And then we took the sequences of there and built an immune library from there. Additional to that, we used that sequence data to identify the murine CDRs, and we built a synthetic library where we grafted those CDRs onto human frameworks. So we had a couple of libraries that we could then use in phage display coming off our in vivo immunization. For the in vitro discovery, what we did is we just selected 3 or 4 relevant libraries from our library of libraries for planning and screening. And for the in silico part, we worked with this company called Macromoltech, And what they were able to do was to really look at the structure of this particular target and then predict what would be putative binders. And then they delivered us 10,000 antibody sequences of putative binders. From that, we shuffled the CDRs and we really expanded that diversity out into a library of 10 billion variants. So at this stage, we have essentially built libraries that we can then use in our phage display workflow of carrying out panning and screening, and then identifying a set of hits for reformatting and producing in those antibodies in milligram quantities. And then we were able to essentially carry out some primary screening on flow cytometry to look at binding to cells and triage down on what we think is going to be some hits for functional evaluation. So I'm just going to show you some of the data that we saw. So first is this primary screening using flow cytometry. This is just a snapshot of some of the data, but essentially what we took as a hit was anything that showed a threefold improvement in binding over with our target expressing cell line over the parental cell line. And these hits were then further titrated to kind of get EC50 values. But it's at this stage here that we started to work with G.CLIPS, and we got some, mice cells that were loaded with C5a receptor from G.CLIPS. And this is, I believe, a very unusual thing for people to do is SPR on GPCR targets. So for this particular case study, we were able to do that using the G.CLIPS antigen, where we could then, gain on rates and off rates and overall KD values and really rank those antibodies to help us triage down as to which ones we would take through to the functional studies. So that it was not just the FACS data, but also the SPR data that we used to help us get there. Then for the functional assay, what we did is we designed this kind of beta-arrestin recruitment assay. And essentially for C5a receptor, for C5a receptor, when in the presence of the ligand C5a, you get this structural change, which recruits beta-arrestin. And we built this kind of cell line where beta-arrestin is fused to beta-galactosidase enzyme acceptor, and the other half of that enzyme is linked to the C5a receptor. So when beta-arrestin is recruited, you get complement halves of that beta-galactosidase enzyme coming together. It makes it an active enzyme, which can then hydrolyze the beta-galactosidase to a light signal. So essentially what we could do is look to see which antibodies then inhibit this ligand to receptor binding and therefore inhibit beta-arrestin recruitment. You can see some of the results here on the bottom right. What we did is we compared it to an antibody that's in the clinic at the moment, an anti-C5a receptor antibody, and we compared what we were getting off our various libraries to this antibody. And you can see just from doing these kind of naive initial discovery campaigns, we found some antibodies that were pretty on par with what's in the clinic. And so this was really promising information and really promising for us. And we tried to see what we could do to further improve some of these. So we took, I'm just going to summarize the data that we've got on the discovery here. So these are the various libraries. You can see this is the in vivo discovery, this is the in silico, and this is the libraries we picked for our in vitro discovery. We use mainly cell panning strategies at this stage, some protein panning. We were able to find binders to all of them, but interestingly, with the functional assays, the majority, although we found functional clones with each of these approaches, the majority was with our in silico discovery option. Yeah, so that was interesting. And what we did, because some of them were so close to that clinical control, is we took 3 of these forward and optimized them using our Twist antibody optimization platform. So here we look at those lead sequences, and we essentially build an optimization library around those sequences using actually natural human antibody sequences that we have within a database. So each of those were built into optimized libraries, and then we plugged that back into our phage display platform, where we carried out some cell-based planning, multiple rounds of this. And at this stage, we had access to the G.CLIPS antigen, and we used that in our later round of planning for this particular clone here, so TB639040. We used, of course, information on enrichment and sequence enrichment, as well as some ELISA data to really identify which hits we wanted to progress with. Again, reformatted them, produced them, and then ran them through primary screening and functional evaluation. Now, this is the stage we're at the moment. So the data is not fully available, but hot off the press is just a very quick look-see at some of that functional data. We've been able to titrate some of these are at this stage not full titrations. We're in the process of carrying out full titrations. But for that clone, where we had a planning strategy where we used cell-based planning and then came in with the G.CLIPS antigen as well in the later rounds of planning, we can see that we're finding quite a number of hits with improved kind of improved clones, essentially. So very promising. There are a number of ways in which we worked with G.CLIPS. So initially, we worked with G.CLIPS to carry out SPR on that particular GPCR. We've used the G.CLIPS antigen as part of our optimization to really carry out some panning. And also what we did is we took some of these hits and leads and we sent them to G.CLIPS for some functional assays. And so that's something I'm hoping Rosie will cover in her case study. I'm quite excited to see that information as well. But essentially, you know, there are just a number of ways in which you can work with Twist. If you're doing your own discovery, you can dip into some of the products that we have to support discovery, or if you wish to kind of partner with us and let us do some of the discovery for you, then just to say we're a really experienced team. You know, we've taken over 400 discovery campaigns and carried these out successfully. We've got over 200 different partnerships. We know of 4 antibodies that have actually been moved into the clinic from ones we've discovered within Twist Biopharma Solutions. Perhaps there are more. And we're using, you know, these 3 different discovery platforms, either individually or in combination, to really give us the best chance of success. So I'm going to stop there. Thank you. And I'm happy to take any questions.
SPR.
Yes.
I'm not sure to have understood what you meant. Did you use it for validation or for orthogonal screening?
So we've used it. Yes, yeah. So the question was on SPR, do we use it for validation or for orthogonal screening? It depends on the project. We have used SPR sometimes to help us triage down. So we'll collect that data and then we'll use it for triaging down. And then sometimes we use it for validation as well. So, you know, when it comes to the lead stage, we will repeat it and ensure that we're seeing the same kind of affinities come off it.
And what kind of throughput do you have?
So we can run hundreds of antibodies in parallel. It's on a plate-based system. So that's how we've set it up to say even within our production system. Our aim is to make sure that we can keep that funnel wider for longer. So take a number, hundreds of antibodies in parallel, make them in parallel, and test them in parallel. So yeah, I say that, so routinely we're running about 96 to 192, I would say, on the Cartera at the moment.
I was quite surprised by the fact that most of your hits were coming from the machine learning. I guess
yes.
It's because already you said an antibody between the hits and maybe paired algorithm
It's a very good point. And yes, I'm also a little bit of a skeptic on that side of things. So I was also very surprised. The details of what macromoltech have used in terms of structures would be based on things that are out there. Interestingly, so we took those sequences that they provided and built this library, and the functional clones we found were actually different sequences, so what in silico discovery is really helping us with is just narrowing down on where to search, and then we're able to kind of find hits within there, and then of course do some optimization to really further in and really home in on that. But yeah. Sure. Cool. Well, thank you. Yeah.
I have one last question that we can discuss it later. I was wondering on your beacon system.
Yeah.
It has to be cells that you use expressing the antigen, or like antigens can be used as well.
Yeah, it's a really good point. So I think that the team's also very experienced, not always using cells. So I think in the past they have used other forms of antigen.
So there's no limitation like the size of the particle.
I don't think so. I think we've been able to use many different sizes, but I guess my technicalities kind of stretch this far. We can have those conversations with the technical team and see what the limits are. Yeah. Great. Thank you.
So we are a little bit in advance, so for the coffee break, which enables us to do some networking around the coffee. So thank you, and let's go back for 11. That would be nice, 11:15.
Allô, allô?
Écarte-toi pour voir que ce soit pas celui-là qu'on prenne.
Est-ce que...
Le problème, c'est que si tu as le micro comme ça, effectivement, là, il peut ne pas entendre.
Mais il faut maintenir le micro comme ça sur le menton
Oui,
et ça transmet... Mais il n'y a pas de retour sur les enceintes. Donc, effectivement,
oui, tu as l'impression qu'il marche pas.
tu as l'impression qu'il ne marche pas.
Ça va être celui-là. Ça va être quand même, hein.
À chaque fois,
Non, non. Il est activé le micro.
il va se connecter.
Mais là, en fait, il y a déjà 8 participants sur 2.
Oui, mais je sais pas.
Parce que là, tu vois, il y a de la modulation d'entrée. Alors, c'est celui-là là-bas, on entend.
Mais si moi, je parle ici dans le micro, la modulation, donc du fait qu'il y a de la modulation, logiquement, on devrait entendre. Après,
Ah oui, mais on n'entend pas parce qu'il n'y a pas le retour, c'est tout.
Et ici, il n'y a pas de retour.
Ouais, c'est ce que j'essaie de dire, mais est-ce que Mehdi, est-ce qu'on peut voir qui est connecté? Les participants. Voilà, si Mehdi est connecté. Mehdi est connecté. Ok. Tiens, vas-y, demande. Ah, d'accord. Si tu peux lui demander ce qu'il veut encore.
bonjour, Monsieur Shelby, est-ce que vous m'entendez?
Peut-être parler dans le chat.
Vous pouvez mettre un message dans le chat.
Il faut parler. Il a son micro d'allumé. Nous vous entendons
non.
très bien. 1h33.
Non,
Ah, c'est maintenant? Ah, ok,
je
merci
voulais
beaucoup.
qu'il m'ouvre, en fait.
Merci beaucoup.
C'est maintenant?
Non, c'est bon, c'est bon, c'est bon. Ah, d'accord. Ok, je vais juste regarder.
Maintenant, 10h33.
Ah oui.
Depuis le début, moi, je suis perdue.
Tu entends?
Alors, il faut qu'il y en ait. Voilà. Allez-y.
Oui, bonjour. Est-ce que vous m'entendez?
Non, et là, je vais faire le test ici.
Si je parle maintenant?
Ouais, là, j'entends. Et là?
Et là, est-ce que vous m'entendez?
Non.
Ok, donc c'est
C'est,
ce micro marche et celui-là là non.
mais celui,
Ah ouais.
mais c'est curieux parce qu'il y a quand même de la modulation quand on
Oui,
parle.
je
Hein.
suis d'accord, mais tu vois.
Oui, oui.
Et c'est que, à force de tomber, il a été cassé.
Mais ça se voit qu'il ne marche pas parce que normalement.
Non, mais il y a pas de-
Ah, il n'y a pas le, ah oui, il y a pas le retour. Ah, d'accord, ok. Ah, ok, d'accord. Ok.
C'est uniquement en local.
Vous pouvez répéter, moi?
Ici, est-ce que vous m'entendez?
Oui.
Normalement, c'était précisé qu'il y avait. Mais attends, là, est-ce qu'on est toujours en ligne? Bon, ben écoutez, on va mettre notre micro et
On va mettre l'autre micro USB et puis on verra si on va faire changer de celui-là. Ceci dit, quand tu vois qu'il est plat ici, c'est peut-être pas normal. Donne-nous le signal, donc au bout d'un moment, qu'est-ce que tu veux?
Okay, so just before the coffee break, Rana talked to you a little bit about the characterization they made by SPR and other assays that use our antigen, so the C5AR in myself. As the Rana mentioned, some of the antibodies that were candidates selected by Twist were sent to us at G.CLIPS. And the idea was to validate or to profile a bit those candidates and see what kind of mechanism of action they can do, how they behave, and do a little bit also of target validation on these antibodies. So what I'm going to show you now is some of the studies we have been doing. This is still ongoing work, so I will show you a few things that we have been doing. So first of all, I'm going to show you how the receptor behave without any ligand added, so at basal state. So we tested actually C5R conformation in these three different lipidic composition and environment, and with all the buffers that we have selected before. And this is the results that you will see here. But before presenting to you this result, I wanted to show you a little bit how our assay works. So basically, what you will see is that on here, what you will be looking for is really the structural change that happens upon activation or inactivation of the receptor. So it's a common feature for G protein coupled receptor that the transmembrane domain 6 will open or close upon activation or inactivation. So the idea is to put a small fluorescent molecule at the end of TM6. This fluorescent molecule has a emission spectra that will vary depending on the environment. So when you activate the receptor, the TM6 will open, and this small molecule, fluorescent molecule, will be surrounded by the hydrophilic or polar environment around it, and then its spectra will change. And the other way around, when the receptor is inactivated, the TM6 will close, and the fluorophore will be a little bit buried in a cavity inside the receptor, and so the emission spectra again changes. And this is what we actually can follow, as you can see here. So this is at basal state. You can see that we have two different J mixes, two different environments, and a shift in lambda max that we can quantify and say if we have open state or closed state, so more active or inactive receptor. And so coming back to the results that you can see here, so what you have here is the lambda max shift, so the shift in fluorescence. And here you have the different condition and different J mixes that we have tested. And as you can see, even at basal stage, you can see that depending on the J mix that we will be using, we will have different stabilization of the conformation. So you see, like for the first one in SB1L7, for instance, you have very active state or stabilization, very good stabilization in the active state of the receptor, which is completely inverse in J mixes SB2L7 and SB2L10. So this is why I think it's really important to make sure with what kind of antigen you will be working for, because of course, a receptor that is stabilized in more active state, like it's the case here for SB1L7, will be more or will have more affinity to all agonistic ligand or antibodies. So if you are looking for, or if you want to select for a candidate with an agonistic profile, it's always better to go, for instance, with an active stabilized state. So we took a few antibodies that were sent by Twist, And so you have them here. We didn't have the same names as you, Rada, so I don't know exactly the conversion of these. But basically what we did is actually we used the antibody. So what you see in blue is actually before adding the antibody. And what you see in orange is after treatment with the antibody for 20 minutes, no, half an hour. Yeah, for half an hour. And we just look at the shift. We followed actually the kinetic, but we didn't have the time to analyze all. So we will have the kinetic curve soon. But at least we wanted to say to see at an endpoint, which is 30 minutes, what happens. And so here's what you can see actually. What you can see is actually when you add the antibodies, depending on the antibody that you would use, actually, you have different profiles. So first of all, here you have the positive control with the peptides C5a, which is an agonist, the natural agonist of the receptor. And as expected, you have a positive shift after 30 minutes addition of the peptide. So it's an antagonistic and activation happens after addition of this peptide. You see as well that the negative control, isotype control, you don't see any difference between before and after addition of this antibody. The same is for these two clones, but some of them, when you added these antibodies, what happens is a decrease in the activation or a shift towards inactive state of the protein. And that's it's the case with this antibody here and this antibody. And that indicates actually an inverse agonistic behavior of these antibodies actually. So not only they do bind, but they do bind and inactivate the protein. These antibodies, they do bind, but they don't seem to have any effect on the receptor, at least when they are alone. So then what we did, because we wanted to go a bit further, again, I'm showing only the endpoints, but kinetics are done, and we will need to analyze all the stuff. So the idea was to see what happens if we first add the antibody and then add the C5 peptide. Do we have something inhibited, or it works well? I mean, we wanted to see how it does work. So we two endpoints. So first of all, we had the receptor without any treatment. Then we added the antibodies to see if they block or not. And finally, we added the peptide of the natural ligand to see how it varies. So here in the first one, we couldn't make any measurements in towards the end because sometimes if we have precipitation in the well or something like that, the measurements can be difficult. But what we see for sure is like for this antibody, that ends with 196. We didn't actually when we added the antibody, of course, nothing happened. But when we added the peptide, nothing happened as well, meaning that the antibody prevented the peptide from doing its job and activated the protein. So most probably, this antibody here is blocking the site of recognition of the peptide. And so then we did the same actually with the others. And as you can see here, this antibody ending with 200, we could still see some activation after addition of the antibody, although the activation is less important from what you have seen before. So in this slide, so somehow it is preventing, but somehow also the blocking or the competition between the antibody and C5 is less effective than in this case, and similar results were shown with this antibody. We also tried to do it the other way around, so first adding the C5 and then the antibody to see if there's competition between both. And as you can see here, for instance, you see that addition, so if you first activate the protein adding the agonist, C5, the addition of the antibody 196 doesn't have an effect. And it's similar for the 201. So what we can say from this experiment, that first of all, there's no agonistic antibody from the ones that we have received. Second, there's some of them with inverse agonistic behavior, as shown before, and some of them can block, of course, the binding of the C5 if they are added first, but none of them so far, at least, seems to compete once the ligand is there. So those are preliminary results that we are doing, but a lot of other experiments are ongoing and planned. Basically, what we also want to see is doing dose response of these antibodies, either alone or in first alone and then in presence of C5. But also doing some kinetics, or the kinetics are done and really follow how the activation and inactivation work, and see actually if we have a difference in the efficacy of blockade between the different antibodies that we have. So that was to wrap up on the case study on C5, and now I'll be moving to another case study that we have been doing. The target this time was a chemokine receptor implicated in inflammation, and the aim was to validate validate the target and profiling of the lead candidate. So the story behind this project, so our client, our partner, had a lead candidate, an antibody with phenotypical results, but they were not able in any cases to show real binding to their target, neither in ELISA or in SPR or in any kind of experiment. They were trying to do so using like usual antigen that you can find in that you can buy. But so they came to us, they said, okay, look, we think that the target is this one. We can see it in vivo or in cellular, and we need really to validate that it's this target and to know a bit what our candidate can do. So first of all, what we've done is a similar workflow as before. So target expression, target expression, target extraction, and stabilization. And then we had this lead antibody candidate, and we started validating. So just to give you a hint about this receptor. So this receptor is a GPCR that is implicated in resolving the inflammation. And the idea was to do target validation and to characterize benchmark ligands and candidates on target in different environments. Because we were thinking maybe the fact that the antibody is not recognizing the antigen that was bought by the partner, maybe it's the folding of the protein that is not the one necessary for this recognition, and which is different from what you can find in the tissue or the membrane and the cells. So again, we did expression and we did the transient transfection in mammalian cells. We had very good expression. We purified the receptor in J mixes. It was pure. We had it mainly in a monomeric form, and so we use that receptor for further validation. So the first set of data that I'll be showing you is after membrane solubilization, actually with J mixes. We tested this MAB1 and a control MAB2. So MAB1 is the lead candidate. It's a conformational antibody. And the MAB2 is a control antibody that recognizes linear epitope. So we reconstituted, or we reconstituted the receptor in different mixes and different lipidic composition. And we tested via ELISA the recognition by MAB1 and MAB2 towards this antigen in all these different mixes. What you can see actually is that in some cases, you can clearly see, like here, for instance, the two recognition between the conformational and the linear antibody are similar. But in some other cases, like it's the case here, you have very huge discrepancy between recognition by the linear epitope and the conformational epitope, which means that if you use this kind of antigen for your profiling, nothing's gonna work, of course. But you will need to use something that really make sure that the conformation that you're looking for is there. So, for instance, this kind of conformation. But also we have also chosen others that were controls and used them for next steps validation. So in the next step validation, we use different also selected environments in which our receptor was reconstituted, and we did dose response ELISA on using the MAB1. And of course, we had a negative control protein. As you can see here at the bottom, nothing happened in all these. So if we focus on these curves here, you can clearly see that depending on the mix that we have used and the stabilization state that we have of the receptor, you can see very different actually Vmaxes and IC50 that are there, which really point out the selectivity of the antibody towards specific conformation or specific environment in which it can recognize the antibody. So we selected in this case one of these mix, not necessarily, or this time it was the best one. And what was nice in that case, coming back to your question, is that the lipids that were used in this mix were macrophage mixes, which really make sense if we think about that the overall aim is really to target the receptor in one mix. So we took this mix and we did our assay, our G-validation assay, either with benchmark ligand. So we had agonistic benchmark ligand here, ligand 1, 2, and 3. And we had our lead antibody and the control antibody. And so here in this case, it's exactly the same principle as before. But what you can do is actually do dose response titration, sorry. You do dose response titration, and you look actually at each concentration, how much shift do you have? And so you will be able to follow the activation of the protein at each concentration. So this is the kind of emission spectra you could have, and then you plot them. Of course, you plot the lambda max shift, and you can have this kind of dose response titration curve. And you can clearly see, actually, of course, the MAB1 didn't have any effect or no activation or inactivation, but MAB2 didn't have any effect. MAB1 behaved actually exactly similarly to all the benchmark ligand that we have tested, all agonists, of course, and had a very nice Vmax and NKD compared to those benchmark ligands. So most probably this this antibody that was selected by the client is an agonist, has an agonistic profile, and also it behaved in or it activated the receptor in a dose response manner. So we wanted actually to go a little bit further and see what happens if we use both the ligand and the MAB, because we wanted to say if there's competition actually between both, if the MAB will disable the natural ligand to bind or not. And so what we did is first to add the ligand, the natural ligand of the protein, and you see you have an activation curve. And then we added MAP2 or MAP1. And so we treated 30 minutes with saturating concentration of ligand 2 and then added MAP1. And as you can see, actually, we have a very nice overactivation of the receptor, probably saying that MAP2 will not bind on the orthosteric side of the receptor, and it will actually act a little bit as a positive allosteric modulator, overactivating the receptor over ligand 2. Whereas MAP1, the control map, you can see that the line stays flat, meaning that it doesn't have, again, any effect on the protein. So this was very interesting and very useful for our client, because first of all, they knew a little bit more about the mode of action of their antibody. They validated the fact that their antibody really acts on this specific target. And they had also now an idea about the KD and so the efficiency and the efficacy with which their antibody can actually activate their receptor. So that brings me to the end of my talk. And so final conclusion and take-home messages. What I wanted to show you today is that G.CLIPS technology can really enable your drug discovery or antibody discovery in many different ways. Of course, we can use our antigens for immunization in a specific format and in specific conformation and environments. But we can also help you doing antibody discovery on targets, either with immune libraries or with synthetic libraries, or maybe soon with Beacon, if possible. And and the final way we can really help you doing it is really by characterizing not only the target validation, but also profiling your candidate of interest. And we can also, so for some of our projects, we did all of it actually in the same time with a lot of back and forth. And this is a little bit how it works with our partners at Twist, with whom we have very, very nice communication on this project. And we hope to continue developing together future therapeutics. I would really like to thank the great team members that I have, more specifically Mehdi, co-founder and associate, and all the other, actually all kinds of different things that you can have in a startup. Mehdi can do it. Thank you so much. Thank you for your support on day-to-day basis. And of course, a huge thanks to Eliciane, Florian, Lila, and Rachel, who's not on the picture, who are really very dedicated team members. Thank you, girls.
I have two questions. The first one on the lambda max shift that seems to be discrete values. And so I was wondering how you explain the intermediate, if it's an intermediate activation.
Yeah, this is actually all. So in a G protein coupled receptor, you have you don't have only one state of activation actually. So the TM6 will start opening and then will be fully opened in some real conditions. So when you, like in this case, not even actually, you can have even higher. So I had some examples in my first slides. I'll try to find them.
Du coup, mes meilleurs résultats, c'est 50% de expected product. C'est pas ouf propre, j'ai genre une impureté qui est polluant. J'ai quasiment plus 5 mètres. Avec le sodium carbonate à 60 degrés.
So see here. Here. So what we have tested here is actually, first of all, treat with nanobody 80. So this is the adrenergic receptor. And you have first treatment with nanobody 80, which is a nanobody that acts like a G protein. So it would pre-activate the receptor, and then you add the agonist, and then you overactivate. What you can see actually is that when you add the nanobody, you have different kind of pre-activation. This means that, I mean, actually to be totally activated, the TM6 should be really open to make actually to the G protein to come and fixate. So sometimes it's really opening, sometimes it's in between. But what you can see, interestingly, is when you add the agonist, everything joins there. So that's the maximum activation that you can get.
So my second question is related to how can you be sure here that what you see will be 20% sensitivity to 30%, and then 100%, that you don't have two populations, the ones that bounce to the, I guess you're using excess of the ligands.
Yeah, it's always on when it's kinetics, it's excess of saturating. And normally we have we have a new microphone. So normally you have we always in kinetics we always use saturating concentration and dose response none. But you have in your dose response, normally you have the maximum shift that you can have at least with a ligand. But what is for sure is you will never have total activation with the ligand alone. For a GPCR to be totally activated, it needs its ligand, but also the G protein.
According to would it be possible to use CDK to see differentiate between agonists and biased agonists?
Yes. So this is something that we're thinking about since long time ago with Guillaume, actually. And the idea is to couple actually in our assay, to couple adding different kind of G proteins like GS or GI or GQ or beta-arrestin, and see if the behavior of a ligand differs. And this is really something that we want to set up. It's not that easy to have access to purified G protein, but I'm counting on the airman team.
But if you know a question, the classical drug discovery pipeline includes at some point structural implementation of the target, etc. Do you think something that you or your clients or police whatever are going to wish to
That's for sure. That's for sure. And this is something that we are trying to establish with different partners. So some of our partners that we are now trying to start establishing collaboration with are either people who are doing X-ray or EM, cryo-EM, or also for epitope mapping, mass spectrometry can be very interesting. So far, we don't have anything settled, but it's for sure something that is of huge interest to us. So that's also a call of partnership. If you are doing any structural characterization, we really would love to test something with you. So yeah, that's something of a huge interest, and because actually it will also help us understand the binding, how it happens, and on which exact epitopes, so on, like what we see in the last case study. If we had something that allows us to do some structural or that give us some structural information here, we can then be sure that the binding of the mAb is really not on the binding pocket of the ligand of interest, which would be another validation or verification of what we think, the conclusion from our assay. So of course, it's something that would be very much needed.
So just for my question, it worth the efforts because you have all these technologies and
It's worth the effort because the information that you would get from this is different from what you get from our assay. So our assay will never tell you where exactly your antibody is binding. That's not possible. It will provide the antibody, but we will need further characterization. But that's also why I was thinking about mass spectrometry, because if you do some cross-linking and epitope mapping, that can be really a very nice way to spot the epitope that are. But Julia, we need to talk.
So, thank you, Bruce, for sharing the data on the C5a receptor project there. So I was just interested to know, so we provided a handful of antibodies which you tested and provided all this information. What is your throughput? Because sometimes we may want to go function first, functional assays and proving
Yep.
the one side.
Yeah. It depends. If you want to just start with like a one shot, like one endpoint, we can screen like we do our experiment in 384 plates, so you can screen 384 at once, but with one point, with one ligand.
Yeah.
And so no, not with one ligand, 384. So with one condition item. But if you want to do kinetics, then it would be more like a subset of candidates that you have already selected, like 10 or 20. How much you do at max? I don't know.
Yeah, like
Maybe around 20.
for like experimental limitations of time and pipetting, I would say 15,
Yeah.
10.
Yeah.
We received about 16.
Yeah, so that's for like those responses and kinetics. It's a little bit more. Yeah. Okay, thank you very much. So I think I'll leave the floor to Julia.
Are you already sharing?
No.
For the pointer.
It's on the
No, but I mean, it's
on the
great.
screen.
I need something. But, right, anyway, use the mouse. Okay, so thank you for invitation. And I'm very glad to be here. It's been very interesting so far. So today I'm going to be talking about antibody characterization and a little bit on ADCs as well, using mainly mass spec-based methods. And so this is going to be an overview of the different techniques that you can use. So here at the IPBS, we don't work a lot on antibodies, but yeah, I made some kind of bibliography and I worked in the past on antibodies. So the idea would be to show you what can perform. So in the lab, we use a lot of bottom-up proteomics. I'll explain you later what this is, but this is classical shotgun proteomics that you do on complex mixture like cell lysates. And after trypsin digestion, you will identify thousands of proteins in a complex mixture. So we use it to identify, for example, binding partners after immunopurification. Or we also use it on purified complexes for QC to make sure that the proteins that we identified, that we only have the proteins of interest in the tube. Then I'm going to be talking about top-down proteomics, where, as you will see, we don't digest proteins, so you have information about the mass and the proteoforms of the entire protein. Native mass spec, which is another flavor of mass spec, where we try to maintain non-covalent complexes in the gas phase to get information about stoichiometry, ligand binding. For example, mass photometry is something that we implemented in the lab recently, and I'll show you some examples of how this can be useful. And HDXMS, so we mentioned epitope mapping just before. So that's the kind of technique that we would use for epitope mapping. So information about accessibility and dynamics, of course. So mass spectrometry is used quite a lot now for monoclonal antibody characterization. So this slide I took from a review from a lab in Strasbourg, where they've been working on antibodies for 20 years now. So they have a big expertise on antibody characterization. And so these are the different techniques that can give you orthogonal information about your map of interest. So as we will see, you can get information about sequencing, conformational stability in the gas phase, glycosylation profiles, accessibility using HDX is hydrogen deuterium change, or FPOP, which is another covalent labeling method that I'm not going to develop today, but it's based on oxidation with reactive oxidative species that will preferably covalently label residues that are accessible. So then, of course, the idea is to label your antibody, your antigen, and then the mixture, and the epitope should be protected from labeling. Right, so antibodies are actually a massive analytical challenge for us, mass spectrometrists, because of so many modifications that you can have. So doing QC on this kind of drug is different from aspirin, right? Where you have just a tiny molecule. So here are listed, not all, but some of the modifications that you can encounter when you work with antibodies. So you can have some differences in sequences, of course, many kind of modifications, glycosylations that are very important. And these will be affected by the cell media culture, or if you express this in heterologous systems. And it's very important because some of these glycans can be pro or anti-inflammatory. So they have to be characterized. You can have also some C-terminal clipping of the C-terminal lysine, for example. You can have some different disulfide bridges, some scrambling, depending on the isotype of the IgG that you're using as well. So all of these need to be characterized before the drug can be approved. And when you're doing a large-scale purification, you need to check that you have the same protoforms, batch to batch. And then you can add another complexity when you're looking at ADCs, antibody-drug conjugates. When you add these payloads, cytotoxic drugs that will bind to different sites, you need to determine what's the drug-antibody ratio of your drug products. So there are different strategies to tackle these questions. The one that's called a TAP, or top-down, is when you look at the entire 150 kilodalton antibody. But then you can try and cut it down. So you can use, of course, reducing conditions, and then you will just be looking at the light chain and heavy chains of 20, 25, and 50 kilodalton. But you can also use what is called, in the middle down approach, you can also use different enzymes that will cut the antibody below or above the hinge regions here, that's shown with these disulfide bridges. And this gives you either the Fab and FC parts of 50 kilodalton, or the FAP2 and 2FC2 parts. And then you can further reduce it in order to get these 25 kilodalton species. And then, of course, in any case, you can digest with trypsin, let's say, and get smaller pieces, peptides, that can then be characterized with a proteomics. And so that's what is used for peptide mapping or glycopeptide mapping, but also for HCP characterization. So that's not something I'm going to be talking about today, but characterization of host cell proteins that are very important for mAb development. So I'm going to be starting first with analysis of mAbs in denaturing conditions. And in this case, this is middle down, meaning that we are looking at the light chain and also at the FC2 parts. And so you can see here the chromatographic separation of the different chains. And in this case, we're looking at an ADC with different numbers of payloads, and they can be nicely separated on this reverse phase chromatography. And then each one of the peaks will give you a mass spectrum. So the mass, you will get the mass of the chains, and then you can select and fragment them in the gas phase in order to do sequencing. So this is just to show the interest of using different fragmentation modes. So we have different modes of fragmentation in this kind of instrument, for example, with electron-based techniques and UV-based techniques. And these graphs show you the nice complementarity between the different fragmentation modes. And you can see here the nice coverage, sorry, that we obtain when we merge all the data. So this is not just one single analysis, right? It's a different analysis and everything is merged. The idea here was to localize the drug, the payloads. And so most of the fragments, they do not carry the payload. And some of them, for example, this one, carries the payload, but not this one, meaning that the drug in this case is localized on this system. Then you can select the ADC that's doubly conjugated with the drug, and you identify these two systems at the conjugation site and so on. So it's a very evolving, fast evolving field, and there's always new advances in terms of technology. Recently, this technique called proton transfer reaction allows you to have even better sequence coverage. I'm not going to go too much into detail because it becomes a bit technical, but just to tell you that this is still a fast-growing field. And these are different type of applications from different labs. This is a work from Strasbourg where they did the same, trying to characterize the ADC conjugation at the middle level. Some labs are, this is a lab from Jenny Broadbelt in the US, and she's expert in UVPD. And in this lab, they used another technique called FEMS, which is high mobility, that helps you to increase the sequence coverage. Some labs also try to look for internal fragments that are gives you even an enhanced sequence coverage. But yeah, I'm not going to go through all of these, just to show you that there's many labs that are very huge interest, because it's a big market, of course, for map characterization and sequencing using top-down and middle-down approaches. So now I'm going to be talking about native mass spec. So as I said in the introduction, the concept here is to maintain non-covalent complexes in the gas phase. So it's kind of a challenge, but it works. So this is what you get in reducing, denaturing conditions. So in acidic condition with organic solvents, the protein will be unfolded. So it will be protonated quite a lot. So you will have high charge states for monomeric species. If you switch to a buffer at, let's say, pH 7, that's compatible with the MADREC, you can get this kind of spectrum for the same protein, right? In this case, this protein forms a dodecamer, so you will get different charge states, and you will get directly the mass of the dodecamer. And this has been applied to many different systems, up to megadalton assemblies. And this is also compatible with membrane proteins, mainly in detergent mice. And now it used to be something quite developed in a few labs, but now most of the instruments that are on the market have this option to give you a native mass spec compatibility. In terms of application for maps, well, the first one that you can think of is looking at the glycosylation patterns. And here it's a comparison between the originator herceptin and 3 different biosimilars. And as you can see, the glycosylation profiles are slightly different. And you can really see that using native mass spectrometry. It's particularly interesting. Why do you want to maintain non-covalent interaction? So some ADCs target cysteines, so you will break the disulfide bridges. So you need to maintain non-covalent interactions in order to see the entire ADC. This is now looking at antibody-antigen complexes and the stoichiometry of binding. So in this case, they studied the map 6F4 that was known to bind to this JAM-A antigen, and JAM-A is actually able to form dimers. And what you see here after titration of the antigen to the antibody, you can see some new peaks appearing corresponding to different stoichiometries of binding. And so here, because JAM is able to form dimers, you can see up to 1 antibody bound to 4 antigen. And then in this study, they compare the binding with the humanized version of this murine antibody, and they could see a very similar trend, showing that the humanization did not affect the binding stoichiometry. In these examples here, they were looking at the deglycosylation and what they could see for trastuzumab, for example. In this case, was the partial deglycosylation. The same for bevacizumab. So this way you can control how effective your deglycosylation is. And in this case, they actually had some partial clipping of the cystaminol lysines. So instead of having 1 peak, you have 3 peaks corresponding to different truncated versions of your antibody. Charge variants are also very important because deamidations can occur. And so we're talking about just 1 delta mass difference on the 150 kilodalton protein. So it's quite challenging. Historically, it was monitored using cation exchange coupled to UV. You can see here the different profiles for different maps. But when you link that to a mass spec, on top of that, you can get the mass, of course, of each one of these species. And you can see here very nicely the separation of these 3 species, even at very low abundance. So that one has no clipping, that one has 1 lysine removed, and that one has 2 lysine removed. And you can actually nicely see that the removal of this charged amino acids is actually changing the charge state distribution of the antibody. And yeah, this is just showing the result for the other antibodies. And you can see that each one of the species could be characterized in terms of lysine clipping and deamidation. Another thing that's, well, this slide shows you the importance of resolution. And here people were looking at mixtures of different antibodies. So this is very specific application, I guess. But you can see here on a QTOF, which is the first generation of our instrument, that the resolution is good enough to separate, let's say, 9 peaks out of 10. If you switch to arbitrage that are more resolutive, you can at very high resolution here, you can differentiate all the different maps that are eluting from the column. And this is just the same with 15 different maps. You can see they are all resolved here at the highest resolution. And this is just another application from Strasbourg on similar separation of multiple antibodies. Coming back to antibodies, I mentioned the measure of this drug antibody ratio. The idea, of course, is that, well, first you need to characterize each one of these species, because all of these species will have different cytotoxic effects, different half-life, probably different release of the drugs that are highly cytotoxic. So you really need to characterize that. The gold standard method is HIC, hydrophobic chromatography, but it can be coupled to native mass spectrometry and ion mobility. And here, just to show you how complex the spectra can be. So in this case, the ADC was analyzed in denaturing conditions, meaning that you have very high charge states. So everything is packed in the low M over Z range of the analysis. So if you select a few peaks, you can still get some drug antibody ratio, but because of the overlap, it's not optimal. After deglycosylation, you simplify the spectrum, as you can see here. And what we did here was to directly go to native mass spec. So you see that you reduce the charge state from 44 to 25, and we compared acquisition on QTOF and an Orbitrap. The QTOF is in red here, and on the Orbitrap, you can readily see all the glycosylations. And after deglycosylation, you simplify the spectrum, but there is still a bit of overlapping. So then what we did was to go to higher resolution, and we added another trick here. We doped the sample with 20 millimolar imidazole, which even further charge reduces the species in order to have a full separation of the peaks and a better estimation of the average star. So native mass spectrometry can be coupled to ion mobility. So the concept here is that in the gas phase, the ions will go through this drift tube, which is filled with a neutral gas, and the protein interest will collide with the gas, and this will slow down the progression of the proteins. And so the bigger the protein, the slower it will go through the drift tube. And so you can separate species of a similar molecular weight, but with different size. And so what you measure is the drift time, and then after calibration, you can translate that into collision cross-section, which is in angstrom square, and it's kind of the average shadow of your protein that's tumbling in the gas phase. Unfortunately, IgGs do not maintain their conformation in the gas phase, but what you can do is increase the energy of these molecules in the gas phase in order to unfold them. And what you see here is what is called collision induced infolding. And so here, basically, you will see the drift time or the size after ramping the energy. So as you will see, you go from a compact conformation to something that's more unfolded, that's an intermediate state, to another unfolded state. And of course, the idea then is to compare different samples. So in this case, they compare different IPGs, and they could show that they had different patterns. In this example here, they selected different intermediates when making an ADC, and they compared the gas phase stability of the intermediates of reaction using this method. You can also compare different glycosylated forms. Here they were comparing the same antibody after deglycosylation and then glycosylation and then reglycosylation. So this is when you want to change the glycosylation pattern of your antibody. And so they compared the one that was fucosylated and the one that was not fucosylated, and they could identify some differences in terms of gas phase stability. Right, so I mentioned already native mass spectrometry. Sometimes it doesn't work. So this is an example where you see the IgG. So the IgG are usually giving very nice signal, but sometimes the antigen, because it's too heterogeneous, will give a spectrum like this, where basically you don't really see anything. And after binding of the antigen to the antibody, the data of the complex again cannot be really exploited. So there are two new techniques that came up recently that have lower resolution, but that will give you a nicer signal. One is called charge detection MS. And you can see here that we not only see the antigen very nicely, but also the complex, and this gives you information about the stoichiometry of binding for the complex. The other one is mass spectrometry that I mentioned already, and you can see here very nicely how much of your antigen is bound to your antibody. This is an application of mass spectrometry. So in this case, they wanted to characterize this bispecific antibody. You can see the main peak corresponding to your drug product, but people are actually more interested in what's in the grass, these tiny shoulders that you usually don't look at and try to ignore. But this is very important when you develop a drug like this, because these tiny aggregates can be very immunogenic. So you actually zoom and try to see what's in there. And in this case, they thought they had some side products of this bispecific antibody. And when they collected the fractions, they could see that they had different activities. And then they used mass spectrometry. So one big advantage of this technique is that it's in solution, and also that it's very sensitive. We are talking here about concentration range in the nanomolar range. Whereas the native mass spec is in the micromolar range. And so they could characterize that this fraction here was corresponding to a dimer of the bispecific antibody. And then you had also some other byproducts of the synthesis. Then using mass spec, native mass spec, you can get the exact mass, and you can see here the main species and then different glycoforms. And I just added that during the coffee break for you, Rosie. This is what we obtained in the lab when we use this technique for nanodiscs. So this is the MSP protein, and this is when we quality control our nanodiscs. So you can see some objects around 120, 150 kilodalton. So now the next step, as you know, is to try and see if we can see a shift when the protein is embedded. Yeah. Next, hydrogen determining change. So I already mentioned the concept. We're going to be looking at the incorporation of deuterium on protein of interest after dilution in deuterium. And the idea is that the rate of exchange will depend on the accessibility to the solvents, and also the hydrogen bonds implicated in secondary structure, and pH and temperature as well, of course. So you will deuterate your protein alone, and then in complex. So in this case, just think that this is an antibody, this is an antigen, and not a proteasome. And after, so again, the idea is that if you have a binding, you will protect the deuteration, so the deuteration will be slower. So after labeling, we quench by dropping the pH and the temperature. Then we do a pepsin digestion, and we monitor the incorporation of deuterium at the peptide level. So for that, we have in the lab this robot that does all the sample processing, and then we inject the samples on the mass spec. This is just to show you one example. So these are the peptides that are monitored during this analysis. And you can see here in two different conditions, this peptide that's protonated, non-deuterated. And after 30 seconds of deuteration, you will see a mass shift corresponding to the incorporation of deuterium. And this way you monitor the kinetic of deuteration in the protein alone, and in the complex, you see here less deuteration because there is protection. So why is it interesting for antibodies? So this is coming back to the JAM-A antigen, and here people were looking at the epitope with different antibodies. And you can see here colored the regions that are protected by the antibodies. And so some regions were common to different antibodies, but some of them were very specific to different antibodies. You can do the same on the antibody, so looking for a paratope. and we're looking now at the mAb NCD1.2 when it binds to the peptide of the antigen CD20, and you can see colored here the regions of interaction. So you would do the antibody alone and with the antigen. You can also follow deuteration on the whole antibody. This is just different time points of deuteration. And then what they did here is comparing the deuteration with before and after deglycosylation. That's deglycosylation. And so they could identify regions that were more accessible when you remove the glycans next to the glycosylation sites, but also regions that were less deuterated, so protected after deglycosylation. In this work, I think that was very nice example. They were looking at an antigen from Neisseria meningitis, and then they took blood from patients that were vaccinated after vaccination. So the idea is that the patients will develop some polyclonal antibodies, and they extracted the polyclonal antibodies, so a mixture of different antibodies, and they did the deuteration with and without the polyclonal antibodies. They could see some protection on the mixture, and they could identify different hotspots, and that are summarized here. And then they use some kind of in silico approach to try and spot the residues that are the most accessible at the surface, and they try to correlate his accessibility with the epitope that they identified. In this case, they compared different. Well, again, sorry, what was it? Yeah, they compared the innovator, so the first drug that was on the market, with the biosimilar, and they tried to see if there were differences in terms of deuteration. They could only spot very, very tiny difference. So the conclusion was that there was no difference with the biosimilar, which is good when you develop a biosimilar. And that's all. Any questions?
Another technical one is not to be precise, to discriminate.
Well, it depends on the size of the sperm that you put in an analysis. So right now, Ron has been trying with we have this project where we insert peptides, and we don't see any mass shift, but the peptide is very small. So we have other projects with bigger proteins, but they don't fit in the nanodisk. So, but yeah, you can if you have a, let's say, 30 kilodalton mass shift, you should see it.
So a G protein coupled receptor would be perfect.
It should work, yeah. So I think we tried with the lithium, but it didn't work out. But we should try again.
Yeah.
We have more experience now with the instruments.
So I had a question. So hydrogen deuterium exchange works on nanodisk in your opinion, and so you can do the experiment without any trouble, knowing that you have the MSP around and so on.
Yeah. I mean, we published some work on a BMRA ATP transporter. Yeah. It's of course less easy than for soluble proteins, but yeah, it works. The hard trick is to get rid of the lipids. So
you need to get rid of the lipids.
Yeah, after labeling. So we label in the nanodisk, and then we disrupt the nanodisk by having some sodium cholate to explode the nanodisk. But that's all in quenching conditions, so at low pH and low temperature. And then we trap the lipids with zirconium beads, and we inject the peptides. I mean, we inject the protein on the pepsin column, and then on the chromatography.
And so you think that if we have lipids around the protein, it would be complicated to do other kind of mass spectrometry? I'm asking the question because I think some of our partners try to do mass spectrometry with our antigen and an antibody. And actually, they couldn't really digest the sample because it was kind of protected with the lipids.
Yes.
Is this something that you have already
Yeah, I mean,
Explain.
usually you get a lower sequence coverage on the hydrophobic part, on the TMs, the transmembrane domains. So there are a couple of tricks. Sometimes you don't see coverage, but it's not because the enzyme is not able to cut. It's because the peptides are very hydrophobic. So you need to use, we use different color. We use a C4 instead of C18. That's binding less to hydrophobic peptides. And this way we could increase the sequence coverage.
So it can be also a technical issue on the
it is, yeah.
Okay.
And then you need to try different digestion conditions. You can use urea, guanydium chloride to really unfold the protein. You can use different enzymes, but yeah, it needs a bit of optimization. Yeah, okay. But people have been doing HDX on GPCRs.
And one last question I had: how? One, it will depend on the technique that you'll be using, but what are the amounts that you would need?
So
like
for
hydrogen deuterium exchange.
Yeah, so hydrogen deuterium exchange, you will need per condition 150 microliters at, let's say, 20 micromolar.
Okay.
Native mass spec 20 microliters at 20 microliters. Mass photometry, what? 5 microliters at 40 nanomolar.
Yeah, so it's really.
It depends on the technique, yeah. And for top down, 5 microliters at 1 micromolar. It really depends, but HDF is the most demanding. That's when you want to do a full kinetics. So that's relates to what the question you had previously. But if you want to increase the throughput, you can when you know the system very well, you can just do maybe 2 time points instead of 6 in triplicate. You can adjust your experiment.
So, SDX experiments, what sorts of times can you reach?
Yes.
Or maybe separate?
So with the setup we have here, the first time points that we monitor is 30 seconds. And then we go up to 30 minutes. You can go higher, of course, but I guess your question was more related to the sub-second. So there are groups developing techniques to do sub-second labeling. There's different ways. You can use microfluidic devices to do a quench flow. And then you can also do sub-zero. You can lower the temperature. But yeah, usually it's the second 2-minute time scales.
Thank you.
Hi all, sorry, you are in mute. We cannot hear you.
Okay, I will just continue, but just saying that the expertise of the platform are distributed on 3 different sites. At the Toulouse Biotechnology Institute, you will find everything concerning engineering and screening of enzymes. At the Laboratory of Synthesis and Physical Chemistry of Molecules of Biological Interest, they have the expertise in synthesis and chemical analysis. And I will focus today on the expertise you find here at the IPBS. It concerns bioinformatics, biophysics, and structural biology, naming NMR and X-ray crystal biology. So starting with the first facility, you will see we have 3 facilities here, dedicated engineers. What they do solution NMR of proteins and their complexes. They have this expertise in protein nucleic acid complexes. They propose also ligand screening. They have a strong expertise in NMR of membrane and membrane proteins. You will find 3 different spectrometers, 500, 600, and 700. They are fully equipped with cryo platform auto sampler, and they can propose solution in solid state NMR. And we have recently developed 2 kinds of ligand screening. They were doing screening using the saturation transfer difference for a long time, but now we have screening based on fluorine molecule, and we have our own fragment library. I will not go in details about the structural biology just mentioned, and focus on the biophysics. We also have X-ray crystallography, again, like for the NMR, the facilities are connected to research team. We do crystallization of proteins and their complexes, 3D structure resolution, and we have developed screening in crystallo with, for example, coated plates with your chemical library, and we can do co-crystallization and solve the structure of the complexes. We have 2 crystallization robots, one for the visualization, one for the crystallization, and more than 2,000 crystallization conditions already prepared. And we have access to synchrotrons, 2 in France and 1 in Spain. So now I'm reaching the purpose of my presentation today, which is the biophysics facility. We have developed this facility, especially the last years, with 3 different purposes. We have biophysical methods for the characterization of your samples and control quality. I will develop that later. We have equipment more dedicated to the screening. Marseille is training of chemical libraries, and we have increased the throughput. We will not reach the throughput we saw this morning, but still we can talk about high throughput since we're working on plates 384 wells. And we have also techniques to measure the binding affinity and kinetics of the interactions. Starting from the equipment for the characterization of your sample, or let's say assessing the molecular mass. So we have 3 equipments that are based on light scattering. The first one is the dynamic light scattering. Rosie talked about that this morning. You will work with just few microliter of a sample at micromolar concentration, non-labeled, and the information you will get, you will access to the sizing of your molecule. You will see aggregation, and also you can check, follow the stability of your sample. I will give you examples for that. And complementary to that, we have the SEC-MAPS. The SEC-MAPS is the size exclusion chromatography, which is coupled with the multi-angle light scattering. This means that in that case, you purify the different molecules you have in your sample, and in line, you have the light scattering, which will measure the molar mass. Now you're not on the estimation, like for classical SEC, where you just estimate the mass based on calibration curve. So how this is useful for your project? So as Julien said, we are not specialists for antibodies, but let's say they are large macromolecule like we have, but I tried to find in the literature examples that they will talk to you. In this study, for example, they use SEC-MAS during the process of production of monoclonal antibodies, because we know that when you produce these antibodies, you can have different oligomers, but sometimes you can produce variants. Of course, you can purify them, but in this study, they were interested in what kind of variant I form during the production process. So they use SEC-MAS. So this is the classical output you will have. In green, you will follow the UV reading after your size exclusion chromatography. And in red, you have the light scattering. What you see here is 3 different peaks. This peak on the right has a molar mass that has been measured of 150 kilodalton, and that was expected for the monomer of the antibody. The peak on the left here has a molar mass of 300 kilodalton, and this is expected for the dimer. But they have this mass between the two, where you have narrow here, it's 200 kilodalton, and they had no idea what kind of antibodies it can be. So what they did is that they purified the 200 kilodalton species, they purified the monomer, they digested them. Thanks to Julian, who presented the different process of digestion. And for the monomer, what they got the mass corresponding to the Fc domain, and they had the mass corresponding to the FAB2. And they did the same thing with the peak at 200 kilodalton. And what they have seen there is that, again, they have the mass corresponding to the Fc, but the mass, the remaining part was not corresponding to the FAB2, but it was larger. And they had an adduct. If you see the difference of mass, they had an adduct of 50 kilodalton. And finally, what they have proved is that what they produced was this antibody, which was non-covalently bound to a FAB fragment truncated from its Fc parts. Another example now about dynamic light scattering. So Rosie mentioned she used that for the characterization of her membrane protein or whatever complex she tried to do. Here they were working on ADCs. ADCs are known to be to have a tendency to aggregate at room temperature, and they wanted to check what the effect of pH or ionic strength on the stability of these ADCs. So I'm just showing here the results for pH. They prepared the ADCs at pH 6.5, and they changed the pH up to pH 7.7. And what they did is they used TLS to measure the intensity of the light scattered and the hydrodynamic radius. And what you can see here is that in all cases, you have an increase of the intensity of the light which is scattered, which is a sign that the molecules are becoming bigger and bigger. And they show that with the hydrodynamic radius, since you can see that as soon at pH 6.5, you have only one peak that corresponds to what you expect for your ADC, while as soon as you change the pH, the size of the molecule increases and it becomes larger than 100 nanometer, which means you really have aggregated form. I told you we have 3 treatments based on light scattering. The third one, Julia already mentioned it, it's the mass photometry. So this equipment has been built by a proteomic platform with speed. He mentioned already that now the advantage is that we are working with just few quantity of sample, with just nanomolar concentration. The difference also is that you assess with the molar mass distribution. It's really scattering, you monitor the light scattering of single molecules while they land on the glass surface. He gave already some examples. This is another example where what they did is that they wanted to quantify the kind of complex they can obtain depending on the ratio. They were using antibodies and antigens. So you see how you can favor just the binding of one antigen or two. And more interestingly, here in this study, what they did, they titrate the association between an antibody and antigens, and they were able to measure an association constant, and they compared that with a published association constant that has been measured by ITC. This is just crazy, incredible. Still talking about stability. We have equipment now that will follow the thermal stability of your sample. So if you're interested in that, we have the classical biorad system where what you do is that you use a dye that will emit fluorescence while your protein denaturates when you increase the temperature. We use that also in routine. You can check the stability of your protein in different buffers with different salts. Also, we use it when we want to see if a small molecule interact with your protein, we know it can affect the TM. It's really easy to set up. More recently, we have acquired the equipment from Nanotemplar, the Prometheus. I know, Rana, you also have it, where in that case, you do not use an external guide. Really, it's based on the intrinsic presence of your protein. You're working in capillaries, and also the information you have at the same time is this back reflection optics. So at the same time, you have an idea of the aggregation of your sample. An example of application of this with membrane protein, in that case, they have this protein, which is a membrane-bound protein, and they wanted to measure the stability of this protein with different detergents in order to choose the best one. What you can see here is that the DDM was the best detergent to use, is the one with the highest TM. And what they did is that they correlate that with the activity of the enzyme. So they measure a kind of TM based on the activity, saying they take the protein, they incubate the protein at different temperature, and they measure the remaining activity. And they have been able to show that with the Prometheus, finally, you found the same results, and it's the DDM that is the best detergent for this product. So I told you about the equipment used mostly for the characterization of your sample. We have last year about this equipment. This equipment is dedicated to screening. And in our case, we use to screen small molecules, but you can screen different things. What you want is really check if two molecules can interact together, and you can measure an affinity constant. So this equipment is based on two detection modes. The idea is that one of the partner will be labeled. Let's say the protein is labeled, right? The first detection mode is what we call spectral shift. Rosie mentioned that. The idea there is that you will measure the intensity of fluorescence at two wavelengths, 650, 670. You will measure the ratio between these two. And what we know is that when a ligand will bind to your protein, it will affect the environment, global environment of your dye. And so this will create a shift. It's really a small shift of your emission spectra. And so it will induce a small change of this ratio. And so if you plot the spectral shift you measure depending on the concentration of your ligand, you can have a titration and you can measure again. The second detection mode they had is still based on this fluorescence. It's not new. It's what we name temperature-related intensity change. This is what was used on the previous machine they had, the microscale thermophresis. While you were working in capillaries, here you're working in a 384 well plate. The idea there is that you will have a laser that will just hit a little bit your sample by 1 or 2 degrees, and this will induce a change in the intensity of your dye. And now this is the end-on state. You do that with your protein alone. And if you titrate with the ligand, if it does interact, it will affect this change in temperature. So this is the gray curves you have between until you reach the blue line, which corresponds to the fully bound protein. And again, if you do a reading at certain time here and you plot the intensity of fluorescence against the concentration of protein, you will assess to the GD. So the advantage of this technique is two detection modes, and you're working with nanomolar concentration of samples, only a few microliters. As soon as the labeling is done, doing a KD is just 5 minutes. So this is 2 examples on tricky proteins to show you how good are the data with this method. So here they were working with a membrane protein, and what they have done here is they use a non-covalent binding between this tag and the dye. And as you can see, they have a beautiful KD measurement. And here it's also another tricky problem. It's IDPs. We know how difficult it is to work with IDPs. Here they were interested in watching the interaction between an antibody and different variants of this IDP or fragments of the IDP. And easily they were able to measure the KD, show that the antibody recognizes the two IDPs the same way, the two variants, but does not interact at all with fragments. I will finish with the gold standard in terms of binding affinity and kinetics, really complementary to SPR. In that case, you're working with non-labeled protein, not immobilized. The problem can be the amount of protein you will need for your titration, and what you're measuring is absorption or release of heat while two molecules interact. And this is an example of application with an antibody, where here they were interested in how an antibody recognizes two different glycan chains. For this one, you can see that you have particle titration, good KD. For this one, the second one, of course, the binding is lower. But more interestingly, what you can get from the ITC is this thermodynamic profile. And we know that in drug development, this is really something you have to consider. And here, for example, for the first glycan chain, what they have shown is that enthalpy and entropy are really favorable for the binding of the glycan chain, while for the second glycan, entropy was not favorable, or worse, it can be unfavorable. Okay, so my presentation, the purpose was to show you that depending on your question, you really have access to complementary biophysical methods. And also it depends on the level of information you want, because you don't need the ITC for all your experiments. And in that case, mass photometry can be really useful because, like I said, it's non-labeled, it's nanomolar, or you will need to label your protein. And you can cover a large range of affinities. And I'm done. I will just finish by this slide showing the website if you have you're interested in the techniques that are developed, or if you want to know more about the platform, and I will be happy to answer your questions. Yes,
I have a question concerning the mass spectrometry, sorry. I was wondering how homogeneous your sample have to be able to show increase in mass when you add a molecule. And my second question is, what's the size limitation of your ligand whenever you want to detect the change in your mass?
Okay, I will start with the second one. The limitation, when you're working with mass spectrometry, the sample you're looking at should be with a mass higher than 30 kilodalton. Lower than that, you will not see it. And the difference of mass is supposed to be 5%, something like that.
5% of the mass of
right. It's the difference between the mass you're measuring and so this is the math. The first question was how homogeneous it has. It's okay. The difference with dynamic light scattering, what you have is really the distribution. So if you're not, what do you mean? It's difference in conformation. No, the peak will be larger. Let's say the peak will be larger. So of course this is some kind of noise, but here the precision of the mass is very good. I don't know what Julian thinks about that.
Yeah, as you said, I mean, by heterogeneous, I guess you mean.
Well, sometimes we have in our micelles we have the micelles with the protein, but of course we have the terminal micelles as well, or J-mix micelle, and those that doesn't have the same molecular weight, obviously.
Well, you should see two distributions.
Okay, so you really see the distribution and how separate they have to be.
Yeah, as actually said the 5%.
Yeah.
If you want kind of baseline or to see a separate peak. Not you will just show shoulders.
Okay. Also, it depends on the level again of information you want. You can imagine peaks can overlap, and you're just looking at the top. Yeah. Or if you want to see if you have interaction, probably you can just look at the top.
The software does a fitting of the Gaussians.
Okay. Yeah.
Quantification.
Yes.
Mass photometry is suitable for screening between a protein that's higher than 30 kilodaltons and its ligands.
Okay. In fact, it's what I show is they were able to titrate the binding of two partners.
Okay, but how many ligands? How many different ligands?
Oh, okay. So when you're working with the mass photometry, you have this glass, and you can do 6 experiments at a time. So for screening, again, I mean, yeah, if you have 1 ligand, why not?
Yeah.
If you have more,
Okay.
Just spend more time.
we have other techniques.
That's a new version of the math complementary with the robots for automated screening.
And where are we getting?
Some business engines.
There's a really nice talk there. Just having a thought about how this works with antibody therapeutic, I guess, discovery and development, you know, what we tend to find is that the detailed kind of assays that sometimes you're showing here are done at the back end of discovery. So as you kind of in cell line development, you have your candidate. But there's more and more interest really to bring some of these further forward in discovery and to use that as some of the triaging methods. So where do you kind of see this going, and what do you see the current kind of limitations?
I would say for the dieters, for example, probably can be complementary to the SPR you're doing, since you're working in high throughput at 384, and probably they will improve this technology, and you can probably work with more molecules at the time. The reading is really fast, like I said. I think reading a plate is about 40 minutes. So that's why I say you can do that with training, since the time you need to fill the plate is really minus, but really, the reading is not. When I say KD is 5 minutes, the KD is really 5 minutes. So probably this technology can go there. I know
another advantage is that you are in solution. You don't need to code or to absorb every protein. That's what we're working a lot on this kind of technologies that with baked, because then you don't need your antigen or even your antibody to be coated. Just mix them in solution and do your measurement. And that's the only bottleneck I would say is really good labeling.
Yes.
Either your antigen or your antibody. But this is something we're working on. Yeah. Cool.
Thank you.
Few minutes and we'd like to free. Some words, concluding remarks. I will not be too long. You have seen today that we need a village to develop a drug, right? So it was a clear example today of the small part of the village, of course. So there is a obvious link between all those technologies, from the idea, the antigen we want to target, the disease we want to target, and the characterization of the drug product, which is at the last end also but useful. All those things are very useful and important as for pharma biotech companies to convince regulators first, because we are aiming at treat patients, investors, internal teams, and to choose the right candidates for the right target. And the challenge for membrane protein is quite huge, but with all together with all the technologies we have seen today, hopefully we are moving forward this pathway all together. So I would say that it would be a concluding remark that make the link between all the presentations, aiming always at treating the patient and find new solutions for those patients who are needed some solution, and we are part of it. So I would like to thank all the speakers. Great job, a lot of preparation. Thank you so much. All the organizations, Twist Bioscience and Biopharma Solutions, feel free to contact them, to reach out to them for any question about all the technology, and it's impressive, amazing. G.CLIPS, of course, for sure, and IPBS also for their platform solutions, and they have a lot of things to bring on the table to bring solutions and answer to questions, because we are answering to questions, and what we bring is information at the end that will be in a dossier that will be studied by regulatory authorities and hopefully brought to patients. So Everything has been recorded, slides will be made available. And again, feel free to reach out to any of the people today that speak out. And thank you so much.